Welcome to solverfy.
For this question, we are looking at the trip operon. And of course, trip here stands for the amino acid tryptophan. And that's one of the essential amino acids that organisms are going to need to live and thrive. So inside of the DNA structure, typically of bacteria, where this trip operon will be found, it's going to consist of multiple parts. You're going to have the promoter region. You'll have the operon that controls this gene on and off switch, and then you have several different trip genes, such as TRPZ, TRPY. They're all responsible for making that tryptophan molecule. So how the trip operon works is there's going to be a repressor bound to this operon here, and that's going to act as the off switch. But this is only going to be bound in the case that a tryptophan molecule is bound to this repressor. With the presence of this molecule here, the RNA polymerase that is going to attach to the promoter region is not going to be able to proceed with transcription due to this repressor and tryptophan complex blocking it. The reason that tryptophan has to be bound to this operon for it to be repressed is because if the cell already has plenty of tryptophan amino acid, it does not want to activate these genes in order to create more tryptophan, because that would be needlessly using energy to create these proteins. However, if tryptophan is not present in the cell, it's no longer able to bind the repressor, and the bacterium has need to create tryptophan in order to survive. So in the absence of tryptophan, the repressor is going to become unbound from the operon, and that's going to free up the mobility of our polymerase to move on with gene transcription. So here the point of the trip operon is to bind the operon in the presence of tryptophan. And it's going to unbind the operon when there is no tryptophan present. So here the best answer choice for this question is going to be A, that this operon works by the repressor binding to that operator and blocking RNA synthesis when tryptophan is present.